Genetic association analysis from the type 2 diabetes research community has discovered hundreds of sequence variants, or SNPs, that are significantly associated with risk of developing type 2 diabetes. Many of these are in or near specific genes. These associations can give us clues about which genes and pathways are important for type 2 diabetes, and that knowledge in turn can help us to better understand and treat the disease. But when genetic associations implicate hundreds of genes in type 2 diabetes, how do we prioritize those genes for further investigation? Anuba Mahajan and Mark McCarthy at the University of Oxford have developed a heuristic for evaluating the evidence that a particular gene has a causal role in type 2 diabetes. It evaluates and scores genetic, regulatory, and perturbation evidence to generate an overall classification of the likelihood that a gene is a type 2 diabetes effector. Using this method, they've generated predictions for each of 132 genes. Now, this gene list and its supporting evidence are available via the Type 2 Diabetes Knowledge Portal. To access this list, select the View Predicted T2D Effector Genes button on the homepage under Explore by Phenotype and click Go. The top of the page gives you some general information and a link to comprehensive documentation about the heuristic and the table. It also contains an interactive graphic that explains the basis for each gene classification. We'll come back to that after we take a look at the table. The default view of the table contains 5 columns: the classification of the gene name, linked to its gene page in the T2D-KP, and 1 column each presenting the overall score from each of the 3 evidence types: genetic, regulatory, and perturbation. The columns are sortable, and the help buttons in each header explain the contents of the column. Now let's dig into the evidence behind the classifications. The diagonal arrows on the right side of each header expand the column to show individual types of evidence. We don't have time to explain each evidence type, but we'll summarize them briefly. All the details are in the downloadable documentation. Genetic evidence considers the significance of T2D associations of coding variants within the gene in 3 large recent studies, and also previous evidence from the literature that mutations in the gene confer diabetes-related conditions in humans. Click on a cell with a magnifying glass icon to see the data behind it, drawn dynamically from the T2D-KP. Regulatory evidence considers whether there are T2D or glycemic trait-associated non-coding variants that affect expression of the gene in T2D-relevant tissues, such as pancreatic islets, fat, muscle, or liver. This category also includes evidence from chromatin conformation studies that a T2D-associated variant physically contacts the gene in pancreatic islet tissue, plus evidence on chromatin accessibility, glucose regulation, and other focused studies on gene regulation relevant to diabetes. Perturbation evidence considers what happens when expression of the human gene or its model organism homologue is blocked. This includes an RNA interference study in which the human genes were silenced in cell cultures, and also any diabetes-related phenotypes arising from mutation of model organism homologues. Within each of the 3 evidence types, the weight of available evidence is combined into an overall score for that evidence type. See the documentation for details on this scoring. Then, the scores for each evidence type are combined into an overall prediction about the gene, shown in the leftmost column. The combinations of evidence types that give rise to these predictions are shown in the interactive graphic at the top of the page. For example, the only way for a gene to be classified as causal is to have a combined genetic evidence score of 1C with a confirmed causal coding variant. In contrast, there are several different ways for a gene to be classified as moderate. It needs to have some evidence from each of 2 types. Click on each of the classifications to see the criteria for types and levels of evidence behind that classification. We plan to update this table regularly and to develop automated methods for collecting and processing the supporting data. We're very interested in feedback on this heuristic. Is your favorite gene missing from the table, or is a gene included that you think shouldn't be there? Please let us know.